Welcome to this explainer. Today, we're diving into what is arguably one of the most monumental scientific achievements in human history. We're talking about the ultimate journey to decode human life itself. Just imagine trying to read a book, but the text inside holds the literal secrets of how you're built, how you function, and even how you heal. Well, that's exactly what we're going to explore right now. So let's establish a clear baseline here. The Human Genome Project, or the HGP, was this incredibly ambitious, massive international research initiative. And the core mission? To actually map and sequence the entire human genome, literally identifying all of our genes. But it wasn't just about reading a bunch of DNA letters. It was about fundamentally understanding the very genetic basis of human biology, health, and disease. It was a real game changer. Here's the path we're taking today. First, we'll look at the blueprint of life, followed by making the map and what's actually inside the human genome. Then we'll cover some real-world genetic revolutions, dig into the ethics and challenges, and finally wrap up with the future of DNA. Let's get into it. Section 1. The Blueprint of Life, the quest to read human DNA. The crucial takeaway here is that scientists weren't just looking at random bits of biology. No, they were collaboratively working to construct a comprehensive foundational reference map. Because think about it, before this project, navigating human genetics was kind of like trying to find your way across an entire continent without a map. You'd be totally lost, right? This initiative aimed to draw the borders, the highways, and the cities of our biology. It was building the map that would absolutely revolutionize genetics and modern medicine as we know it. Section 2. Making the Map, the timeline of a scientific marvel. Okay, the timeline on this is actually pretty mind-blowing. The journey officially launched back in 1990, and by the year 2000, scientists were already announcing the very first draft of the human genome. Then, in 2003, they hit a massive milestone. The main goals of the project were completed amazingly 2 full years ahead of schedule. But, science never truly sleeps. It wasn't actually until 2022 that the remaining, highly complex, unresolved regions were finally completed by the telomere-to-telomere consortium. That finally gave us our very first truly complete human genome sequence. So how did they actually pull this off? Well, to build this map, researchers relied on a highly structured procedure. First up was DNA sequencing, which is basically determining the exact order of the nucleotides. Then came genetic mapping to locate specific genes on our 23 pairs of chromosomes, followed by physical mapping to figure out where those DNA fragments physically sit. But honestly, the real unsung hero here was bioinformatics. They were generating just mountains of data, unprecedented amounts. So advanced computational biology and bioinformatics became absolutely essential to store, compare, and analyze all those complex DNA sequences. Section 3. Inside the human genome. Discovering the astonishing data. 3.2 billion. Just let that staggering number sink in for a second. That is the amount of DNA base pairs that make up your personal human genome. To put that into perspective, if you were to read out every single letter of your own genome at a rate of 1 letter per second, it would literally take you over a century of nonstop reading just to get to the end. It's just massive. But here is where things get really fascinating. Hidden within those 3.2 billion letters, scientists found something completely unexpected. They identified only about 20,000 to 21,000 specific protein-coding genes spread across our chromosomes, along with the mitochondrial DNA we inherit from our mothers. Why is that surprising? Well, before the project, a lot of scientists assumed human complexity would require upwards of 100,000 genes. Turns out, we do a whole lot more with a whole lot less. And that brings us to one of the biggest surprises of the entire project. It turns out that only about 1 to 2% of our genome actually codes for proteins, which leaves a vast 98% as non-coding sequences. Now, back in the day, this was initially dismissed as junk DNA, but actually, scratch that. We now know these sections act as absolutely crucial regulatory elements. They're essentially the intricate control switches for how our genes are expressed. Not so much junk after all. Moving on to Section 4, Real-World Genetic Revolutions. Moving from abstract science to tangible impact. So once we had the map, how did it actually change the world? Well, mapping the genome revolutionized a massive array of fields. In medicine, it unlocked pharmacogenomics, which basically means drugs tailored to your specific genes. In biotechnology, it gave us the foundation for synthetic biology, allowing scientists to engineer entirely new biological systems. Then in agriculture, we're seeing things like marker-assisted selection for disease-resistant crops.
And of course, in forensic science, human identification through DNA fingerprinting has completely transformed criminal investigations and paternity testing. It literally touched everything.
The crucial takeaway here is that the project fundamentally transformed biomedical research. And these aren't just abstract concepts. These benefits are actively saving lives today. We now have a vastly improved understanding of human biology, which directly leads to much faster, earlier disease diagnosis. It's opening the door for better, highly tailored drug development, and explosive growth in precision medicine where treatments are completely customized to you, and really powerful new advances in gene therapy. It's a completely new era of healthcare. Section 5. Ethics and Challenges. Navigating the Implications of Our Blueprint. Here is a really heavy question for you. Who actually owns your genetic information? The architects of the project recognized the immense weight of this early on, and that's exactly why they built a dedicated Ethical, Legal, and Social Issues, or ELSI, program directly into the initiative to study these exact implications right from the start. Because when you unlock the fundamental secrets of human life, you face some incredibly complex societal hurdles. If we can read your DNA, how do we protect your genetic privacy? How do we prevent genetic discrimination by employers or health insurers? As a society, we really have to ensure absolute informed consent when gathering this data, navigate the really messy laws of genetic ownership, and maintain strict ethical boundaries when it comes to the use of advanced genome editing. These are massive questions we're still grappling with today. And beyond the ethics, scientists also had to conquer immense technical challenges to get us to where we are today. The project faced incredibly high initial costs, for one. Plus, remember those 3.2 billion base pairs? That massive data generation created an unprecedented headache for data storage and management. And honestly, even today, interpreting the complex functions of that 98% non-coding DNA remains a massive, ongoing challenge for computational biologists around the world. Section 6. The Future of DNA, Where the Map Takes Us Next. The original project really just paved the way for the staggering recent advances we're seeing now. I mean, we've moved from a billion-dollar, decade-long project to next-generation sequencing, where whole genomes can be sequenced incredibly rapidly and affordably. We now have CRISPR-based gene editing, which actually allows scientists to make precise cuts in DNA. We've got single-cell genomics, letting us study biology literally one cell at a time. And we're increasingly using artificial intelligence to analyze vast population databases, giving us a completely new understanding of human health. Ultimately, all these future prospects point to a total transformation of healthcare. Things like disease prevention, AI-assisted diagnosis, and precision oncology, meaning cancer treatments, are going to be entirely personalized to your specific genetic makeup. By integrating global genomic databases with the study of proteins, RNA, and cellular metabolism, medicine is rapidly shifting away from a one-size-fits-all approach to highly targeted, totally individualized care. As we wrap up this explainer, I want to leave you with a provocative thought to chew on. The map has been drawn, the blueprint of human life is laid out before us, and the technology to read it is sitting right in our hands. So, how will reading your own personal genetic instruction manual change your future? Thank you so much for joining me to explore this absolute scientific marvel. Keep questioning, keep learning, and we'll see you next time.